see. Xu Chen, the distinguished professor from San Diego, as well as Geert Schmidt-Schoenbein, and also our speaker tonight, Viviana Gradinaru. So this effort was initiated in order to be consistent with the goals of the Bioengineering Institute of California, which is a University of California Office of the President, MRU, Multi-Campus Research Unit. And what we wanted to do is build bridges between biotechnology, biopharma, that is industries, but also the academics. And not only academics UC-wide, which is what the BIC represents, the 10 campuses, but also pursue something that was sort of bigger and greater than that. And that's why the Bioengineering Institute started the California Academy of Bioengineering, which consists of each member of the National Academy who practices in California and does biotechnology and biomedical engineering. So as the first activity, an executive subcommittee was formed, Geert Schmidt-Schoenbein, myself, and a few others, in order to put together this symposium series. And we're doing it every other month on a Monday evening. And the idea is to present leaders who are the primary thinkers in our field to describe future trends in biomedical engineering and also where they see engineering going, not only their current research, and provide a forum for all of those practicing in academia and industry. And so at this point, I'd like to hand over the introduction of Viviana to Xu Chen.
Thank you, Scott. It's my great pleasure and privilege to introduce our distinguished speaker, Professor Viviana Gradinaru. Dr. Gradinaru received her BS from California Institute of Technology in 2005 and her PhD from Stanford University in 2010. She returned to Caltech 10 years ago and is now Professor of Neuroscience and Biological Engineering and Director of the Center for Molecular and Cellular Neuroscience. Dr. Gradinaru has combined neuroscience, protein engineering, and data science to produce microbial opsins that are tolerated by mammalian cells and viral capsids that are capable of crossing the blood-brain barrier in adult mammals. These products have enabled the high-precision, minimally invasive repair of diseased nervous systems. Dr. Gradinaru studies the mechanisms underlying neurodegeneration, its affected behaviors, and relevant interventions such as deep brain stimulation. She has also developed tools and methods for use in neuroscience, such as optogenetic actuators and sensors, tissue clearing and imaging, and gene delivery vehicles. Dr. Gradinaru has published her outstanding research in some 90 publications in top peer-reviewed journals such as Cell, Nature, Science, Neuron, and PNAS. She has more than 10 patents and is a co-founder and board member of Capsida Biotherapeutics, an AAV engineering and gene therapy company. As a result of her outstanding contributions, Dr. Gradinaru has received many awards and honors. These include the NIH Director's New Innovator Award and Pioneer Award, the Presidential Early Career Award for Scientists and Engineers, the Outstanding Young Investigator Awards from the American Society of Gene and Cell Therapy and the Society for Neuroscience, and the Gill Transformative Award. Dr. Gradinaru is a Sloan Fellow, Pew Scholar, Valley Scholar, Moore Inventor, World Economic Forum Young Scientist, HMRI Investigator, and the inaugural Pierre Gross Young Investigator. Most recently, she received the Science Magazine and Pins Prize for Neuromodulation and the Vilsack Prize for Creative Promise in Biomedical Science. and she was elected as a AAAS Fellow. These remarkable recognitions reflect Dr. Gradinaru's marvelous accomplishments that have made major scientific impacts. It gives me great pleasure to invite Dr. Gradinaru to give her California Academy of Bioengineering's distinguished lecture on targeted non-invasive genetic therapies for brain disorders. Dr. Gradinaru, please.
Thank you so much for the amazing introduction, and I hope I will live up to it. I have to say that this was work from many collaborators and students over the years and mentors. And I need an approval that you are seeing the right rendering.
You just swap screens, it'll be perfect.
Okay, let's swap screens. I want to make sure that we have a good experience, and I can see the chat right now, so that's what I learned from prior talks, if you rely on chat. Thank you so much to all of you for making time to attend at what I heard is an atypical hour, but this is the dilemma of us working parents that have to pick up little ones. So I hope maybe this accommodates a broader audience than usual. So thank you very much for your time. And today I will try to do justice to work from mentors and collaborators and trainees, many of them that you see here. Some of them are alumni that went to better places, but I included a pre-COVID picture to get the sense of the team that did most of the work that I will talk about today. And this work has to do with our efforts as neurotechnologists. So we are neurotechnologists that are trying to invent and use methods in service of understanding the brain and improving brain function. And we've done that in the context of Parkinson's disorder. So these are the circuits that we've worked most on. And these are the circuits and the patient needs informed technology developments that we focused over the years. And they had to do with methods to control neuronal activity with high precision. And this was part of my doctoral work that I continued in my lab at Caltech. Also, ways to, once we can control function, you also need to know the routes where the function is directed towards. So we develop methods to clear tissue, to render tissue transparent in order to visualize circuits that underlie therapies such as deep brain stimulation. And for both tissue clearing, one form of it known as clarity, and optogenetics, we've learned early on when I started my lab at Caltech of significant bottlenecks in gene delivery. Many experimental paradigms rely on transgenesis or gene delivery, and we learned that especially for larger brains or for non-human primate brains, this was a significant barrier. So this is why the bulk of our work and what I will focus on today would be around the gene delivery, but I'll try to give you a sense of how we use this in the context of neuroscience and what motivated our tool development, because it's been a cycle of back and forth between applying the tools, refining them, and informed by the basic science projects to push forward methods that we still need. I think we made a lot of progress, but much is needed still. And I'm looking forward to an exciting discussion period afterwards and maybe ideas and new collaborations. Very briefly, my conflicts of interest, it was mentioned already, and also some of the technologies that I developed both at Stanford and Caltech, they're licensed by the respective institutions. Scientifically, I've been focusing over almost 2 decades now on neurodegeneration. And at first, my focus was on a circuit perspective and very brain-centric. And we've made some progress in applying optogenetics to understand the deep brain stimulation, rather than acting on the cell bodies in target areas such as subthalamic nucleus, they were acting on fibers of passage or fiber synapsing into the area, which complicated the picture of understanding how deep brain stimulation works. And we've pushed some of this work early on with trainees, and we looked at different deep brain stimulation targets. And time again, I developed maybe, and it's fair to call it, a dissatisfaction with the fact that we were looking too late in the progression of the disease. And we were highly brain-centric. And the great thing about being in a broad division at Caltech is biology and biological engineering, is that I have colleagues that think about immunology and think about other organs in the body and emphasize the fact that the brain does not act in a vacuum. And with this in mind, we do know from Parkinson's patients that way before motor symptoms, there's perturbed sleep and there's gastric dysfunction. And this involves a much broader CNS, central nervous system, and PNS, peripheral nervous system involvement. And this is where both of our tools and our knowledge is lagging behind in the PNS exploration and also connecting the brain to the body. And over the years, we've focused on a few technologies. So I just want to give you an overview of methods that I helped develop and we've used with trainees in the lab around labeling circuits and microscopy, high-speed microscopy to image thick, clear volumes. And also, more importantly, methods for functional control, whether they have to do with opsins or the non-invasive modality of chemogenetics, where you don't need to deliver highly scattering and maybe not as expansive light, but you can deliver small molecules to control, again, genetically tagged circuits. And by using these anatomical methods and functional methods, we focus first, for the reasons that I mentioned, Parkinson's as a sleep deficit problem early on, and then gastric deficit. We focused on a few key areas that were involved in sleep deficits and arousal deficits. And we've had two studies, and I want to highlight one of them just to show you how we've been embedding the technologies that I mentioned. But one set of studies was focused on these very special neurons in the dorsal raphe. So dorsal raphe is a nucleus known for its serotonergic content and involvement in the neuropsychiatric spectrum. However, it has a sparse population of dopaminergic neurons, and my CNS graduate student, now graduate and postdoctoral fellow at Princeton, Ryan Cho, showed that these dopaminergic neurons can modulate arousal and promote waking by salient stimuli. And the details are in his thesis and publication, but what's interesting to note is that in humans, these neurons are known to degenerate. So dopaminergic neurons in the dorsal raphe degenerate in multiple systems atrophy, and also in Lewy body dementia. And these disorders are commonly, they're associated with excessive daytime sleepiness. So that connects to the human disorder. Of course, the dorsal raphe is much more often known for its serotonergic population, which is a very robust network in the middle of the brain that has been involved in sleep, but its role has been highly controversial. And this is where my colleague, David Prober at Caltech, he runs a genetics lab on zebrafish sleep. And David had this, based on transgenic fish in his lab, had this theory that actually the serotonergic raphe neurons promote sleep. However, the literature was somewhat confusing for the following reason. And that's when David approached me and said, we need to look at this in parallel in two different model organisms, one for genetics, one for closer behavior to mammals. And we decided to embark on a project that took the zebrafish and mice and try to maybe get to the bottom of it. Is serotonergic dorsal raphe sleep promoting or wake promoting? And the literature, the body of literature has been wide, and its involvement in sleep was clear, but the exact role controversial for the very following reason. Early studies that used lesion, so either a chemical lesion or a mechanical lesion of these serotonergic cells in the dorsal raphe, suggested a sleep promoting function, because when you affect them, you affect the sleep promotion. However, as technology evolved and we gain as neuroscientists the ability to do single cell recording, so the ability to record from these cells, what we learned was that the serotonergic neurons in the dorsal raphe, very importantly, are wake active. So this generated a bit of a conflict. How can wake active cells induce sleep? And so again, different modalities, and we set up to address this in two model organisms with all of the genetic tools that are strong in the zebrafish and all of the physiology tools that we had in the lab for rodents. And first, we went on to confirm that indeed, ablation of the raphe serotonergic neurons reduces sleep. And we use the toxin, so you can see in the intact animal, you have a strong population that we lesion with the toxin. And then we had, by we being former graduate student, neurobiology graduate student, Michael Altermatt, had EEG recordings and ENG recordings to very quickly label the states of sleep. And Michael was able to confirm what was known in literature from lesion, non-genetically targeted lesions and chemical inhibition, that indeed ablation of the raphe would basically reduce sleep. And these are the recordings based on so with the EEG, you can label the wake state, the non-REM, and the REM state. However, we wanted to go and excite this area and try to understand, if you excite the neurons, would you be able to alter the sleep-wake cycle? And this is where things got interesting, because here we were able to use the full power of optogenetics, which unlike chemogenetics has the feature of high temporal precision. So what Michael did was to deliver opsins to the dorsal raphe, an extremely difficult surgical area. And the difficulty is because it sits under the aqueduct. The aqueduct has fluid. Any perforation to the aqueduct, you lose the animal. So Michael perfected this rather difficult surgeries. It's a mouse, it's a tiny brain, and was able to implant 2 fiber optics to the side of the aqueduct and be able to deliver light to these cells deep in the midbrain under the aqueduct. And then ask himself whether optogenetic activation could indeed promote sleep in mice, right? So what he learned, again, by using EEG and EMG to identify the stages of sleep, the wake, non-REM, and REM, was very interesting, because indeed tonic, low frequency activation of these cells would promote sleep, right? So that was consistent with the idea of promoting sleep in mice. However, burst high frequency stimulation induced wakefulness. So a much higher count and accelerated activity would induce wakefulness. And this helped reconcile a bit some of the past contradictory results where stimulation of the dorsal raphe would give apparently not consistent results, but the reality is that all of those experimental results were possibilities that depended on the genetic identity, on precise genetic targeting, and the precise frequency control. And so this brings back the peculiarity still. How can neurons, so what I mentioned was that these cells are wake active, right? But through lesion, and we did the parallel experiments in zebrafish. I'm showing you only the rodent experiments because those were done in my lab. But David Prober's lab did all of the parallel experiments in zebrafish. And we got to the conclusion through lesion inhibition, genetic and physiological, that the serotonergic raphe neurons promote sleep. However, we also, by measuring with GCamp, and I didn't show that, but it's described in the paper, we confirmed that these cells are wake active. So the puzzle is, how can neurons that are wake active be sleep inducing? And this goes back to a beautiful hypothesis that was introduced by Jouvet, saying that cells could act as a counter that builds homeostatic pressure. So the animal, the mammal, is awake, and these cells are adding sleep pressure. And once there's a certain threshold, the mammal can go to sleep. The reason why I wanted to highlight some key findings from this study was precisely to introduce the need for technology development that will follow for actually the bulk, the key part of the talk. Because what you've seen here is how spoiled we are as neuroscientists and bioengineers and as a community when it comes to genetically tractable model organisms, to the zebrafish, to the mouse. We were able to look in these animal models to do genetic ablation, genetic activation, measure the activity with very high precision in behaving animals and correlate with behavior. And this is strikingly different from the needs of larger and non-transgenic brains. And this is where I want to focus the bulk of today, because if we are to go from the mouse, the small mouse brain, to the human brain, we have a few fundamental challenges in terms of methods and tools in order for us to understand mammalian brain biology and to restore function. And this has to do with difficulties in delivering genes and also difficulties in delivering photons. For better or worse, light is a loved modality to both image biological systems and influence biological systems. However, light has very big deficiencies when it comes to large, highly scattering and highly absorbing environments. And this is why for the bulk of my work, I focused on ways that we can bypass these challenges with engineering systemic AAVs that can cross the blood-brain barrier and deliver genes to non-transgenic brains. And also in matching these delivery methods with actuators that are strong enough to be coupled with a non-invasive delivery for non-invasive access and maybe influencing physiology. And I hope I will also have a chance to show how we apply this in the context of neurodegeneration. For gene delivery, there are key features that are needed for safe, effective, and minimally invasive delivery. And we need vectors that are capable of needed specificity. Specificity defined broadly. It could be cell type, for example, inhibitory cells, excitatory cells. It could also be brain region or organ or circuit specificity. At the same time, we need vectors that can bypass very difficult body barriers. And two that come to mind are the placenta, so the placenta to embryo to access the developing brain, and also the blood-brain barrier to access the brain from the vasculature, from the bloodstream. And yet another difficult problem that we are in early days of trying to solve it, and it's a very difficult one, it seems, is the packaging capacity of such delivery vectors. So we focused in the context of my lab on the adeno-associated viral vector, and we are benefiting, we're fortunate to work on it at this time because we're benefiting from 5 decades of intense study of this fascinating moiety. So since its discovery, we've learned a lot about basic AAV biology, about repurposing these AAV vectors for gene delivery, all the way to today where we see approvals such as the one for spinal muscular atrophy, with AAV9 being delivered to young children and increasing their lifespan and motor ability. And it's amazing to see the good that's done. It's also, as a neuroscientist, frustrating to see the difficulties that we still have, especially around the brain and especially around the safety of systemic gene delivery. And to transition in the needs in terms of the brain delivery, as a community, we are used to using different AAV serotypes that have slightly different tropisms for cell types of organs. And we are very good at changing the cargo, packaging the gene of interest with gene regulatory elements for refined pattern. However, we are most times than not delivering these through highly invasive methods. So here I'm showing you in the context of a mouse brain, how we can access deep brain areas, hypothalamic areas. And you can see a few, you can see a lot in this image, but a few things I want to emphasize. One, the invasiveness, so how much matter you have to go through and possibility of blood vessel damage and hemorrhage. But then with the spot injection, you have a limited spread, so poor volume coverage, and you have a lot of copy numbers at the injection site, which could be toxic. And away from the injection site, there's not enough product to be functional. And this is all in the context of the mouse brain. So as a reminder, as we go through from the mouse to human, if we are to use the same method, diffusion rules don't change when you deliver straight to the brain parenchyma from the mouse to the human. So to get the physiologically meaningful coverage level, you need multiple insertions that have poor distribution and side effects. So then one approach we took was, can we instead do gene delivery to the entire brain across the blood-brain barrier instead? The blood-brain barrier is there for a good reason. It protects us from disease, but it also prevents research tools and also therapeutical elements from accessing the brain. This is why Parkinson patients get L-DOPA and not dopamine, because L-DOPA can cross the blood-brain barrier and convert into the effective moiety. How can we cross the blood-brain barrier? So here is where it was extremely helpful to think as an engineer and not as a neuroscientist. And throughout my work, I've had, for better or worse, switch back and forth between these two thinking modalities, which at times was challenging. And it's hard to know exactly when to best apply one method or the other. But in neuroscience, we've been trained to value hypothesis-driven experimental planning and very rigorous control of all possible variables. In engineering, we are used to quickly prototype, right? Quickly prototype and trust the process and aim for intermediate good enough versions that will advance you. So that was an approach that I took with Opsin development to refine the tool, knowing that what we have now, it's still not good, but having a process that can improve the biological or chemical moiety. So this is where the method actually that my colleague Frances Arnold in chemistry, she invented the method of directed evolution that relies on the fact that if you have a starting point that has the property you want, but is not strong enough. So in our case, I mentioned AAV9 that's used now in gene therapy for children with spinal muscular atrophy, can be delivered to the bloodstream, crosses the blood-brain barrier weakly. But in the adult mammal, it's not a very good tool. However, it's a good enough starting point. So you can take AAV9 and then apply molecular diversification and generate libraries of diversity. It could be more than a theoretical diversity, more than 1 billion variants. And then by using directed evolution, in the starting point, and then you need to put all of your thinking and hard and smart work into the experimental design of the selection process. And this selection process, if stringent enough, it allows you to raise above the biological noise and narrow down the 1+ billion diversity into a handful of variants that are worthy of in vivo testing to see whether from your library, starting with an AAV9 parent and with offsprings diversified at variable loops, can you have a capsid that now can cross the blood-brain barrier to reach the adult brain? And the answer is yes. So we took AAV9 from a capsid that very poorly, and it's mainly astrocytes here, very poorly transduces the adult mouse brain. And by applying the directed evolution of the capsid surface, we are able to engineer a gene delivery vector that can transduce the entire brain. And as good engineers, we didn't stop here because the dose required was quite high still. And we engineered the flanking amino acids. And this resulted in a version that's used now by, I would say, hundreds of labs worldwide. And there's lots of publications already and many in the works, where this gene delivery vector is used to deliver interesting cargo. And because we only modify the shell, the cargo is at the discretion of the experimenter. So anything that fits the AAV packaging size can be incorporated here. Through the process of directed evolution, we also obtain some other capsid profiles, such as the serendipitous PHPS that I will tell you about later, how we use it in the context of periphery and Parkinson disorder. However, for the capsid from neuroscience perspective, many times you do not want to transduce the entire brain. That level of non-specificity doesn't serve in terms of hypothesis testing for neuroscientists. Because you have flexibility in the cargo, though, you can package instructions that allow that pattern to be refined, and you can get coverage. So all of these patterns here are throughout the mouse brain with different either promoters or enhancers, or mini promoters or microRNA target sites. that can give a non-invasive expression pattern in areas that are surgically difficult. So remember I mentioned with the dorsal raphe under the aqueduct, you can actually genetically transduce the dorsal raphe without the difficult surgery that's in the proximity of the aqueduct and can involve a lot of side effects. You can transduce the cerebellum, which is a very interesting area from disease perspective and also motor coordination perspective, completely non-invasively. And one of my favorite examples is how much coverage you can get throughout the cortex when you package an inhibitory neuron enhancer. So this can serve not only to get good coverage, but you can package a library of gene regulatory elements, and you can with one injection get the brain-wide profile. The alternative method would be injections in different brain areas, one at a time, to try to understand the profile of a particular enhancer or promoter. Much of biology involves complex cell types connected in complex ways that sustain our complex mammalian behavior. And this is why we already need to think beyond monogenic applications. And here I'm giving you just a proof of concept that you can use the vectors to deliver multiplex cargo to cell types like the myelin or astrocytes or neurons throughout the brain with one bloodstream injection. And one can have the capacity to dial up or down the level of gene transfer. And this is important in a few ways. One way we used it was in terms of connectivity. So you can deliver the 3 fluorescent proteins, green, red, and blue here, and you can dial up and down in the inducer. I'm showing you 2 different animals, the olfactory bulb in these animals. And the way we found just the right dose of labeling, so we can tell apart crossing fibers. So if these were the same color, we would not know they're from different cells in the area. So this can help combine with tissue clearing, because when we try to reconstitute morphology and connectivity, it's very important that we, can cover large volume areas. So I hope the movie is rendering okay through the Zoom. But what you see here is a cleared volume of tissue where we labeled and then we reconstructed the morphology. Of course, these were all examples that are used for visualization, but for gene delivery, the most exciting maybe aspect is the ability to do physiological control. So I want to give you an example of how we used, and this was a DARPA-sponsored project, we use these gene delivery vectors to deliver chemogenetic actuators across the blood-brain barrier. And one advantage of chemogenetics is that the small gating drug also crosses the blood-brain barrier, so everything can be non-invasive. And one can basically just target the midbrain dopaminergic neurons, and you see here confirmation of their genetic identity. And then you can activate these cells and you get the expected behavior based on prior neuroscience research. Of course, you get hyperlocomotion phenotype in the experimental animal, and the control animal is just out and about chilling in its cage. We have similar experiments for place conditioning as well. And this shows that you can use these systemic vectors to modulate physiology in a non-invasive way. So what's important about this experiment is not the finding, because this was known from literature and expected from literature, but is the ability to obtain this behavior that's genetically targeted in a way that involves no surgery. So everything is done through the bloodstream with the retroorbital delivery. There's no fiber optic, no craniotomy. However, as I showed you in the example of the dorsal raphe serotonergic study, and the reason why I like to start with that study is that you've seen the striking difference between tonic and phasic firing. Same area, same genetic identity. If you do tonic versus bursting fire, activity control, you get very different behaviors. And this is the case also for dopaminergic cells in the VTA for reward. And there's many examples like that. So this brings back the need for methods that have high temporal precision, such as optogenetics. And when we first tried to use optogenetics with systemic delivery, we weren't successful because there's one maybe downside of systemic delivery. You get much fewer copies than you would get with a direct injection or transgenic modality. So then you need potent actuators or potent sensors to bypass that more limited delivery of genes. So that's what, and then we had to basically had to go back and work on the opsins to make them much higher conductivity. So then with the same number of opsins on the membrane, we could now modulate behavior through these systemic vectors. And of course, we went back to the same exact method that worked for the AAVs, and that's the method of directed evolution. This turned out to be a much harder problem. And the reason was a much harder problem is that directed evolution works wonderfully when you have throughput on extracting the superior offsprings. But if your method to evaluate, so in AAVs, we had DNA enrichment as our output measure. And DNA enrichment, because of sequencing methods and the scaling now, it's very straightforward to go through very large libraries. However, for optogenetics, the gold standard is still wall cell, single cell patch clamp. That's very slow and very difficult. And one can do it, and our lab is good at this, but even with very good skills and multiple rigs, you can only go through maybe a few constructs a day if you need to have enough statistical power. So this method, what we had to do was to match the high throughput power of directed evolution, to bridge, not to match, to bridge it with the low throughput nature of the way we were measuring the opsins. And for a while, we were wrapping our head around this. How should we go about it? And my former student, Claire Bedbrook, co-advised by Francis, came up with the idea that this can only be bridged by data science. And although I was skeptical at first, Claire managed to convert me, because she built a very clear flow where she would build channel rhodopsin sequence diversity. And she would then put this library through an intermediate higher throughput test, which was a fluorescence test for membrane expressing opsins. So when you modify opsins, some break, some don't make it to the membrane. They need to be in the cellular membrane to modulate conductivity, to either suppress or activate action potentials. And if they're not in the membrane, they're not the ones you want. So Claire introduced this fluorescent readout for membrane based on a spy tag, spy catcher system that she published previously with my group. And then out of the variants that were expressing well, she patch clamped only a subset. So basically from the space, she did detailed measurements only on about 10% of the entire diversity. And then she used statistical models to predict on the dark space, to predict from the ones that she didn't measure, which sequences will display interesting properties in terms of their light spectra or kinetics or conductivity. And when Claire bridged these two methods, actually it worked very well. And I have to give her credit because she started with 120,000 variants, and this is impossible to do with cell patch clamping on it. And by combining getting training data sets from a subset and then predicting on the rest, she picked up 28 variants that ended up with very interesting properties. And I'm gonna, and properties being the ones that in our community we think about when we think about opsins. And I will highlight a few constructs. She called them chargers. And what you see, they have much higher conductivity. So you can look here with direct injection. So this is direct injection of the parent opsin, channel rhodopsin, and then the opsins that Claire made. And then with systemic delivery, and again, the systemic delivery, the problem is that you just don't have as much channel rhodopsin per cell. And in collaboration with a postdoctoral fellow in the lab that now has his own lab at Children's Hospital in Cincinnati, Elliot Robinson, they showed that in a living mouse, you can do non-invasive delivery of chargers. So you can package the engineered opsins in the AAV vectors that cross the blood-brain barrier, and you can deliver the opsin throughout the mouse brain. And then you can just thin the skull and apply the light. So again, there's no craniotomy, it's just thin skull to increase light penetration. And then you can modulate animal behavior similar to how I showed you in the chemogenetics experiment, with the advantage now that you have temporal control. So now I'll transition slightly to how we are using these vectors in the context of disease and of Parkinson. And I showed you vectors that can target the brain very well, and somewhat serendipitously, because these libraries are very large and because we have the ability in the lab, and in the interest of time, I won't present that body of work, but a while ago, we were able to make entire mice transparent, aside from bones. Bones, we clear them later. And by having the rodents transparent, we were able to look broadly with low resolution, but very fast throughout all of the organs. And when we did this, this allowed us quick profiling of the biodistribution of lead AAVs. And some properties we found even though we were not looking for them. And one such vector was the PHPS, that we found it to have a great distribution in the gut, in the enteric nervous system. And this was fortuitous because at the same time, my lab was neighboring the group of Sarkis Mazmanian, and they are experts in how to do histology and investigation of the gut and the stomach. And this informed the next stage of the work that had to do with trying to expand this gene delivery platform beyond the brain. And more importantly, so this is gut delivery, but more importantly, understanding that to be effective in curing the brain, we want to make sure we actually, through systemic delivery, we want to make sure we leave other organs alone. And here's an example of how you might want to avoid liver transduction, and I will elaborate on this part later. But for now, I want to show an example of how we use the PHPS vector, the vector for the gut transduction, to understand something very interesting about Parkinson physiology as it relates to the gut. And this study was performed in collaboration with Laura Volpicelli from the University of Alabama and Sarkis Mazmanian. And the postdoctoral fellow that led it, Colin Callis, wanted to know what happens when you seed the gut with a small amount of preformed fibrils of alpha-synuclein preformed fibrils. And alpha-synuclein underlies a large class known as synucleinopathies, and Parkinson could be an example of it. And Colin wanted to know what happens to the physiology, to the local physiology, and also with how this might cross to the brain and cause brain deficits as well. And this was because of a body of work around tissue from Parkinson patients showing synuclein aggregates in the gut, and also basic research showing that when the gut is seeded with high level of aggregates, one can see pathology progressing to the brain and motor deficits. However, the story took an interesting angle when Colin tried to do that in healthy, young adult mice. And when Colin seeded healthy and young adult mice with modest level of preformed fibrils, it turned out that these mice had some short-term deficits that they could basically recover from just in the gut, and that they could recover from very quickly. And at first for Colin, this was frustrating because he was, based on all of the hypotheses, he was expecting pathology that would progress to the brain. But then he had the brilliant idea to do the same exact experiment in two other cohorts, one aged mice and the other one mice that were overexpressing alpha-synuclein, the Taiwan alpha-synuclein overexpressing mice that are model for Parkinson disorder, made by UCLA professor Marie-Françoise Saleh. So when Colin did this experiment, he learned something very interesting. In aged mice and in the alpha-synuclein overexpressing mice, a modest seeding of preformed fibrous in the gut could develop in long-lasting pathology in the gut that then would cross and evolve through the brainstem and also lead to deficits. And then he wanted to know what are the molecular mechanisms that the same path, a gut injury, would be short-term in the young mice, but gut injury, whether there's a genetic risk factor or whether there's aging, would develop in pathology. And he looked at lysosomal enzymes and learned something very interesting. He narrowed down on duodenal GCase. GCase is a lysosomal enzyme encoded by GBA. GBA mutations cause Gaucher disease, and also GBA mutations are an additional risk factor for developing Parkinson. And what Colin learned was that in wild type, you have strong levels of GCase that they decrease in genetic mutations, so in alpha-synuclein overexpressing mice, and also in the aged mice. And this decrease in the lysosomal enzyme inversely correlated with the amount of pathology, whether in the amount of toxic alpha-synuclein or neurons displaying altered behavior. And then we thought of a genetic rescue experiment where, with the hypothesis that GBA would produce GCase that's beneficial in the outcome of neurodegeneration here. So we used a very fast expressing Q vector system, so packaged GBA under a controller, under an amplifier, and deliver it to the gut. So again, systemic delivery, we didn't need, because of this virus, we didn't need to perform abdominal surgery. So through retroorbital bloodstream delivery, we delivered GBA to the gut. And you can see here in the Western blot that we can restore GCase levels with this system. And after restoring GCase levels, Colin was indeed able to see also rescue of physiology. So when he looked at the gastric physiology and also at transit measurements, that was able to improve outcome. As neuroscientists, we were very interested what happens to the neuronal activity in the gut, because of the hypothesis that any dysfunction would precede behavior and it would precede very striking measurements. So we tried to basically move a lot of the methods we used in the brain for many years to move them in the gut. And that proved to be quite difficult. So for starters, the gut tissue, if you put channelrhodopsin in it and you try to do electrophysiology, when you stimulate the neurons, you get contraction of the gut lining and very severe motion artifacts that make data analysis very, very difficult. So what Colin ended up doing was to do sparse delivery of channelrhodopsin. That's why you see so few neurons here. So then when we activate the neurons, it would not get, so what you need to see here is stable stage for pulses of light, and then you see activity in the neurons that Colin could measure. So then by looking at the calcium waves, so this, we delivered a calcium sensor, and by looking at the calcium level, we could measure that pathologic alpha-synuclein actually affected neurotransmission by looking at the calcium signal. And then when we replaced GBA with the systemic vectors, we could get partial rescue. And partial is important to emphasize here because GCase is just one lysosomal enzyme. Synucleinopathies are very complex disorders, and likely they don't rely only on a single mechanism. But what I think it's important to understand from the study is that it's just a starting point for many questions, and at least for us, showed us how much more work we have to do in order to both understand more from the basic research perspective, but more importantly, to translate these findings. And one area that, and I hope to have time to show you some developments around that, the systemic AAVs are not specific. So bloodstream delivery will reach all of the organs and all of the cell types, and this can be a liability and can have serious side effects. We need better tools for the peripheral nervous system. And importantly, the AAVs will perform differently across species. What we can do in the mouse is not necessarily the same way we'll perform in a human. And we need more disease proof of concept applications for these newer AAVs. With systemic delivery, one can access the entire tissue. And that's why it's important to look at the throughout the body. And we can look throughout the body in two ways: with high resolution in postmortem tissue, or with lower resolution, but in vivo. And for in vivo, we teamed up with a group of Kathy Ferrara at Stanford. She runs a PET imaging lab, and we were able to together label the engineered AAVs with PET labels and look real time in living mice at the biodistribution of these engineered vectors. And what you can notice here is how AAV9, even after a day post-delivery, is still mainly in the liver, while engineered vectors such as PHPEB accumulate and stay in the brain. But what's important to note is that the liver is still a strong expressor. And this can be a serious problem because of toxicity associated with liver delivery. And this is where we go with higher resolution methods to try to understand which organs have in maybe too much biodistribution and which cell types. And this is where the method that I mentioned, clearing the entire mouse, so we can clear the mouse at once and then extract different organs. And we can look at all of them. So you can deliver one or multiple vectors, and you can look at all of the organs and learn that, yes, you transduce the brain better, but you also transduce the liver and the heart, and this could cause serious problems. So this was the reason why we then went back to our selection systems, and we said we need to ask much harder questions. Not only we need vectors that cross the blood-brain barrier, but we need vectors that are detargeted from problem organs such as the liver maybe, or the dorsal root ganglia for pain. And to do that, we reworked our diversification, capsid diversification methods, and we conducted screens in parallel in different Cre transgenics. And we're fortunate that we have Cre transgenics for many different organs and cell types. And we can package a cargo that depends on this Cre recombinase. So this is the stringency in the assay and what allows us to raise above the noise, and also what allows us to refine tropism. So basically at the end of this directed evolution screen, in the tissue, you can only read the barcode that completed all the steps. So for example, crossing the blood-brain barrier, making it into the nucleus of a cell that has the Cre enzyme. Now the Cre enzyme will only act on double-stranded DNA. With AAV is a single-stranded DNA vector. So in order for the Cre enzyme to act, it needs to complete that step of double-stranding, which is a bottleneck usually. So many engineered vectors fail. Cell entry is only one step, but there's many steps after entry. And by having this enzyme acting on the double-stranded DNA, with just one screen, we check for a lot of points along the way with just one in vivo experiment. So then through this process, the result is very large data sets that we look across Cre lines and we look across DNA enrichment. And with this, we were able, and I'm compressing maybe years of work and subtlety here, but we were able to take AAV9, and this slide maybe is like the one that I like the most. We were able to take AAV9 from a vector that transduces the adult brain very poorly across the BBB. It's beautiful in the liver and it's used for liver. To a variant that can cross the blood-brain barrier and transduce the brain, but you still see maybe too much liver expression if the liver is not your intended target. And now to versions of AAVs that can cross the blood-brain barrier and have no targeting of the liver. And this is just the quantification for what I told you. The next immediate step is if we have this capacity to engineer tropic vectors for cell types or for organs, can we actually take it from mouse to other model organisms, and even more so, maybe to humans for therapy? And this is where things become confusing, but confusing is always interesting for scientists. The vectors we select perform the best in their selection strain, which is not surprising. You get what you select for, says Francis all the time. So AV, like PHPB, performs very well in the strain you was selected. Now, if you look at other strains of mice, some of them get transduced and some of them not. But by looking at the genetics, so this is a slide that always gives me pause when I think about human gene therapy trials. These are all laboratory mice, highly inbred. They're all mice, they're just different strains. And this is how a vector engineered to cross the blood-brain barrier performs across these strains. So now if I look at the audience here, we all will have a slightly interesting, right? By the nature of our ancestry or other biological variables, we're going to have slightly different BBBs, and how we design the gene therapies to serve everybody. And the good news is that once you have such striking patterns, you can then look at the biology of the blood-brain barrier and learn the mechanism of these vectors. And indeed, we were able to do that. So by looking at the mechanism and by now selecting in multiple strains of mice, I'm happy to say that we do have vectors that can also can cross the blood-brain barrier in previously restrictive mouse strains such as BALB/c. And more importantly, the same framework could be taken to step now to different strains. And one example is the ability to now cross the blood-brain barrier in the marmoset brain, which is a New World monkey, very interesting experimental model. And we can here engineer vectors that can transduce the marmoset brain. And interestingly, depending on the vector type, you can get slightly different tropism. You can get neuronal biased, or you can get a profile that also captures astrocytes. And this profile is brain-wide. So you can, if you step through the marmoset brain, you can see expression throughout the brain. This is very exciting from an application perspective. And it also tells us that we can ask for these libraries not only to cross the strains, but also look at more refined cell type specificity. And by using the right Cre-transgenic selection and the right data analysis, we can now look through these data sets and try to come up with families of related sequences and tropism. So now we're putting, it's interesting, we took a somewhat agnostic directed evolution approach to come up with a few useful tools. Now that we know they're useful, we can work backwards and ask ourselves, what's the biological mechanism that these tools are using? And then put that knowledge to work in getting better libraries and refining the tropism. And interestingly, from these data sets, you can now select capsids that can have cell type tropism in the absence of gene regulatory elements. So I want to emphasize all of these images are obtained with fluorescent protein delivered with a ubiquitous promoter. However, the morphology is very different. It's neuronal or astrocytic or endothelial just by the virtue of the capsid shell. And this is helpful because now you free up precious genetic material, you put the tropism onto the capsid. And also, you might avoid cell types that could be problematic. If neurons are the target, you don't want maybe antigen-presenting cells like the astrocytes to also get the virus, because that would also soak virus and you will just end up injecting more than you need for the effective result. And right now we are progressing this work, and just a few more minutes before we close for Q&A. We're progressing this work with single cell RNA-seq, and this is a recent study published where we try to be extremely comprehensive in terms of the tropism of these vectors. So we look at all of the cell types that have them and also their impact onto the cells that they transduce. And I want, hope this will play well, just to maybe appreciate why systemic delivery is powerful. If one wants to target the vasculature of the brain, if you don't have transgenic means, you need to inject in the brain matter, but this can damage vasculature and can break precisely the cell types you're interested to study. So what I'm showing you here is a hemisphere of the mouse brain with tissue clarity and image with a light sheet microscope. And you can see the entire vasculature transduced just by a systemic route, by vascular injection. And of course, as I mentioned, now we have capsids that don't require promoters or transgenics in order to get this pattern. And also we are advancing the PNS variants. It's important to, as I mentioned, to think beyond monogenic disorders and also to think beyond disorders that have a single target. And I can't think of a better example than maybe Friedreich's ataxia, where you need very good gene delivery to the brain, but also to the periphery, because there's heart deficits, there's also muscular deficits, and the disorder is caused by lack of function in the frataxin protein. So gene therapy works well because you can just deliver the frataxin gene and make enough of the product. What we were able to do was to combine vectors, the best one for the brain, and then the best one for the periphery, and deliver frataxin to all the tissues in need. And I'm going to show you just the behavioral outcome. And I hope this will play well through Zoom. So this is a pole walking, a beam crossing motor assay. And you can see that the wild type crosses it without any trouble. Then we have the ataxic animal that has a slower crossing and many slips. So you can see the difficulty in crossing. And then with gene delivery, 2 capsids, packaging the frataxin, and I'm going to let this play. You can see a much faster crossing and fewer slips. And of course, this was done in more mice and repeats, and I can show you the summary data. And what we see is that we can have targeted gene therapy with systemic AAVs for the central and peripheral nervous system, and we can rescue motor phenotypes in a mouse model of Friedreich's ataxia. This is very exciting, and of course, it makes one wonder how translatable it is. And I will leave you with a few notes around recent and ongoing collaboration with my longtime collaborator, Dr. Fox at UC Davis at the Primate Center. And Drew and I started a collaboration many years ago where we engineered the vectors and we sent them to Davis for testing. And I'm showing here what I think is maybe the first rainbow-like macaque, where we can deliver genes across the blood-brain barrier in a young rhesus macaque. And there is expression throughout the brain, including in brain areas of high relevance for disease, right? Subcortical areas. These vectors that I talked about are all available on Gene. This is, if needed, a decision tree of things that we've worked over the years, started in 2012 for the mouse, and then most recent marmoset, to rhesus ongoing work, and hopefully one day human cells, for both central and peripheral nervous system, with different tropism depending on the interest. And this is hopefully a way to both understand the mammalian brain better. And I've said a lot. A few key points might be around systemic delivery can be highly beneficial if we minimize the risks, and also if we work on combining it with powerful options such as gene editing. And I'm of course very grateful to the funding sources and my team. You've seen their names throughout. I almost need to defend my choice of not including pictures. I know many PIs choose to include trainee pictures with data. I do not, because when I see a face, I cannot look at data. That's how my brain works. So I always, I include the name and the journals, but I do not have the trainees' faces because I think it distracts from their beautiful data. But this is the group, and I'm very grateful for your attention, and we can pause for questions. Thank you.
Awesome. Song, you want to take it away?
Oh, great. Let me turn this. Great. Dr. Gradinaru, thank you so much for this fantastic talk. We are going to have Q&A sessions. I'm Song Lee from UCLA. I will be the moderator. So in Q&A, there's the first question from Yishao Wang from at the UCSD. I don't know whether you can see the question, Viviana.
I think so. So I can.
The question is regarding whether this. Yeah, please go ahead.
Mm-hmm. So the question is around the mechanism. What do we know about? So if I'll just read it, right? So is it the selected capsid? Is it because the capsid causes a more packed viral particle to allow better permeability through BBB? Or AAV-PHP promotes an active gain of function transport targeting mechanism, or both? Great question. We now know that is the latter. There's not a change in the size. So we did TEM, we did measurements of the capsid. It's not a change in the size that will make a smaller particle. And that's good because a change in size would have also reduced the packaging capacity. Instead, it's an active process. And for PHPEB, the mechanism has been worked out by 3 groups independently and has been found to be Ly6A that promotes transcytosis across the BBB. So the PHP vector binds, gains functionality, binds Ly6A, and then can cross. And now we are working in the lab to, if you allow me the word, to deorphanize the other engineered vectors that I showed. And one more in the chat. If the AAV subtype works in one mouse strain and marmoset, will it work in all mouse strains? Okay, and then I'll read the second one afterwards. The answer is no, not necessarily. The answer is not necessarily. It will depend on the BBB properties. And we've had vectors that work in mouse and marmosets, and we've had the same vectors that work in one mouse type and not another mouse type. So this underlies the importance of experimental testing, one, and second, finding the mechanism to which these vectors cross, because now knowing that PHPEB crosses to Ly6A, we are not surprised that it doesn't work in non-human primates, or that it doesn't work in mouse strains that do not use Ly6A in this way. And then there's a second part to this question. If it works in mice plus marmosets, does it guarantee it will work in humans? And I would add, if it works in mice and marmosets and macaques, does it guarantee it will work in humans? Again, not necessarily. So I think that's why it's important to know the mechanism. Once we have a capsid that's validated, this gets us closer to knowing the mechanism. And then you can check whether the same mechanism is present in humans from, for example, sequencing data or genetics. One more, is cell type specificity concerned across species? Very good question. For some, yes, for some, no. CAPB10 has neuronal tropism in mice and in marmosets. Other vectors we found for tropism in mice do not necessarily have the same tropism in other species. So again, it goes to say how complex biology is, and the fact that we still have to chip at it. And we're fortunate that we have a motivated team and support of the NIH to answer exactly these questions that are answered here. Mm-hmm.
I could jump in with a question. So I'm really quite inspired by the gut-brain barrier study, the gut-brain axis studies that you're showing. And you said that some of the AVs are tropic to both of those, or one's even more specific to the gut. But recently, at least in the immunology literature, connections between the gut and the brain have been known to be, adrenergic and going through the vagus nerve. And so can you ever actually light up that axis between the gut and the brain with some of the AAVs?
We're working on it. So I think that's a very good question, and it goes from people that have vagotomies that are more resistant to developing Parkinson's. An fascinating body of literature. And the vagus, so the difficulty is that it innervates so many organs, right? So that's maybe the one difficulty with the vagus. And what we found, so the difficulty in getting the same resolution for the peripheral nervous system as we are approaching with CNS is the following: in the material that we have for the directed evolution experiments. So you need, when you deliver the library, you go to your target tissue and then you extract the DNA from that target tissue. The less you have, the less your odds for success. And that's why we also failed when we tried to do circuit based AAVs. The AAV retro is a beautiful example, right? So AAV retro works. So we try to, for example, make AAV anterograde, but the less material you have, the harder the problem becomes. So I think the answer is, it's an interesting problem. We're working on it. It might be harder than others that we are working on.
Limited anatomically, huh?
Exactly. And now in the mouse, now again, if we have more real estate in terms of the tissue to access, but then the larger the experimental subject, the less likely is that we will have the Cre enzyme there, because the transgenics are not available. And when you don't have the Cre enzyme, you get more noise.
Okay, I think there's a question regarding the size restriction. So if this is an active transport process, is there any size restriction? It's from Victor Rogers at UC Riverside.
I'm trying to guess the sub-question in it. So it's active transport because of the 7-mers binding onto Lys6A. So now, are those 7-mers sufficient by themselves to carry something bigger? And that would be fantastic, because AAV is small and doesn't carry as much. This was tried. These 7-mers were taken and tried, including our collaborator on the PET study, tried to deliver larger moieties. It turns out that there's something about the AAV environment. It might be because what's maybe not apparent, when we engineer the sequence, you don't get only one 7-mer on the capsid, you get a repeat. And depending where you put it, you can get as many as 60, depending where in the coding region you put it. So we think that these 7-mers might act collectively to dock onto the receptor and cross. So the 7-mers themselves are not necessarily able to carry more. In some cases, we are able to take the 7-mers from one AAV serotype and move it to another AAV serotype. And that's been helpful for, let's say, serotype switching problem if you fight against neutralizing antibodies. Mm-hmm.
Good. Do you want to ask you a question?
There's one more I see in the Q&A. What will be the first human translation of your technology? I am a basic scientist. I'm a professor at Caltech. I'm not a clinician. I'm not in biotech. However, it became obvious that these vectors can be very helpful for patients and for human disease. And that's where I teamed up with talent in the medical space and biotech space that can grow this into solutions for patients. And the first applications will be in the neuroscience spectrum because of my interest in neuroscience and what we develop these vectors for. They won't necessarily be limited to brain disorders. And I think neuropediatric indications are the ones that are most likely early candidates.
Dr. Gradinaru, this was a wonderful talk. I'm extremely interested in your story about the gut. And the gut, as you know, is in different physiological functions during the day, depending on whether we ate something or not. And so I have, for the purpose of this discussion, a very simple question for you. Have you looked at the AAV transfer into the brain or into the liver during different times of the day, when a mouse had something to eat and when it didn't have something to eat?
Fascinating question. The answer is not to this particular situation. We looked whether is night in terms of sleep, because of our sleep work, we cared a lot about the sleep-wake cycle and the circadian rhythm. So we tried to look at different biological variables. We looked at different sexes and we looked at different circadian spots. We did not look on satiated mouse or not. That would be not hard to do. And again, from the perspective of designing gene therapy, I remember one paper reading that one could increase transaction to the brain if you hold the animal upside down after delivery. So just thinking about all the possible variables, right? Whether it's gravity or physiology or that can influence, I think that would be a fascinating thing to do. Maybe I can convince my next rotation student to take a try at it.
It can be
Especially
done by you can do this by have an undergraduate student do that. They will enjoy that.
The best would be to do this in the context of the PET study, because then with some vectors, we were able to see the routing through the gastric versus through the liver. We see some going through the gastric path rather than the liver. And that might be, at least you can know quickly enough whether a satiated system maybe blocks. You could imagine a lot of constriction effects, a lot of interesting reasons why it might differ. Thank you for the project idea. We'll see if I have customers.
I'm looking forward to the answer.
Give me a bit of time. I'll take a note.
Maybe I can ask a question regarding the targeting evolution and also immune responses. So for targeting evolution, when you get these different variants of viruses, what is the mechanism that more accumulating more in specific organ or tissue? And also for different virus strain of these AAVs, do you observe different immune responses, for example, from T cells or macrophages, etc.?
Yeah, so we have, and thank you for the question. With systemic delivery, I think the first concern that always comes to mind is immune response, because with direct, the brain is somewhat privileged, the eye is somewhat privileged, so with direct delivery wouldn't have this. We look, so when we do systemic delivery early on, mice are very, how should I say, robust and tolerant. But then once we saw the BBB crossing for the mouse and we started to design experiments for NHPs, we started to be very careful because we wanted to make sure there's no room for error or getting NHP sick without us thinking through all of the possibilities. So then we started to do measurements of liver enzymes like ALTs, and we see elevation in that at times. And this is also the reason why we embarked on the study I mentioned that's now in Frontiers in Immunology, where we do a 10x profiling of cells in the brain to see what pathways are activated. And we see short-term, so what we see, it's a inflammatory response, like P53 pathways, up to 3 days, and then it goes down. And the same study could be done, it's a matter of skill and cost of going through all the tissues and all cell types, but one could imagine doing the same for liver or the heart. So I think immune response now, it's a big one. And it's not only the T cells, so that's in terms of toxicity, but also the response in terms of neutralizing antibodies that might make this not effective. And that's a different line of work. And I know many groups are working on this problem. It turned out to be a very difficult one. And as we expand gene therapies, I think especially for our age group, children most times don't have much neutralizing antibodies. So I think in the adult population, the prevalence is much higher, and that would make administration more difficult. So then the other question was, how do these vectors get it? We think it's endocytosis. There's the receptors for, there's a series of receptors for AAVs. I think what we do when we engineer the surface is we break some of those interactions and we add others. And by that combination, we did target or retarget. if you want that word. We retarget the tropism. It's hard to be 100% specific. And that's what we found. So many times intersecting the capsid profile with the cargo is still needed to get truly refined specificity.
Great. I think you have addressed all the questions. So I want to thank you for the wonderful seminar, and also thank all panelists and participants. And we will conclude this seminar now. Thank you.
It's my pleasure.
I particularly enjoyed the theme of whether it's hypothesis driven